Hey there, fellow microbe enthusiasts. Today we're taking a deep dive into the world of Corynebacterium, CP.
Exciting.
It is, right? Get ready to unlock some fascinating facts about this bacterial genus. We're going to focus on Corynebacterium diphtheria.
The one that causes diphtheria.
Exactly. And our guide is Principles and Practice of Clinical Bacteriology, the second edition by Gillespie and Hockey.
An excellent choice, packed with information about these clinically significant bacteria.
Sometimes overlooked, though, right?
You could say that?
Yeah. All right, well, let's get started. What can you tell us about the key characteristics of Corynebacterium spirium?
Well, they're incredibly diverse, found in lots of different environments.
Oh, really? Like where?
Soil, water, even on the skin and mucous membranes of humans and animals.
Wow.
But despite their diversity, they share some unique structural features. For instance, their cell walls have mesodiaminopimelic acid.
Okay.
And short-chain mycolic acids.
Wait, mycolic acids, don't those show up in mycobacteria too?
Good catch, like the ones that cause tuberculosis.
So there's an evolutionary link between them.
Exactly. But here's a fun fact. Not all Corynebacterium species have mycolic acids.
Oh, like which ones?
C. amyrcolatum and C. croppenstedtii are the rebels of the family.
Interesting. So they're kind of the exceptions to the rule.
You got it.
That's what makes microbiology so cool, right?
Yeah.
Always something new to learn.
Absolutely.
So besides their cell walls, what else makes them stand out?
Their appearance under a microscope is pretty distinctive. They're usually straight or slightly curved rods.
Okay.
And often arrange themselves in a palisade pattern.
Palisade pattern, what's that?
Imagine a row of tiny pickets all lined up.
I see, like a fence.
Yeah, kind of. This arrangement comes from their snapping division during cell division.
Snapping division, that sounds intense.
It's just a quick separation, which gives them that characteristic V or L shape.
Interesting.
Oh, and they have these things called metachromatic granules.
Metachromatic granules, what are those?
They're basically storage reserves of phosphate, and they look really cool under the microscope.
So they're like little energy snacks for the bacteria.
You could say that.
Pretty smart.
Yeah.
But not all Corynebacterium SPP are bad, are they?
Right. Some are harmless, just hanging out.
But others?
Others are pathogenic, like C. diphtheria.
The one that causes diphtheria.
Precisely.
That's a disease with a scary history.
It is. There's evidence that it might go back to ancient times, maybe even Hippocrates' time.
Wow, really?
Early descriptions, like those by Lomius and Bayeux, describe it as a suffocating illness.
That sounds awful.
It was terrifying, especially before we knew what caused it.
So how did scientists figure out that C. diphtheriae was the culprit?
It was a series of discoveries in the late 19th century. First, Edwin Klebs saw microorganisms in the membranes of infected people.
Interesting.
Then Friedrich Loeffler actually isolated and cultured the bacteria.
That must have been a big breakthrough.
It was. It proved the bacteria caused diphtheria.
But just knowing there wasn't enough, was it?
No, they needed to show how it caused the disease.
And how did they do that?
Well, Emil Roux and Alexander Yersin did this amazing experiment. They filtered a culture of C. diphtheriae.
To remove the bacteria itself?
Exactly. They injected this bacteria-free broth into animals.
What happened?
The animals got diphtheria. It proved that a toxin in the broth, not the bacteria itself, caused the illness.
Wow, that's clever.
It was groundbreaking. This led to the development of antitoxin and vaccines.
Life-saving stuff.
Absolutely. Emil von Behring even won the first Nobel Prize in Medicine in 1901 for his work with diphtheria antitoxin.
Incredible. These early discoveries were game changers.
No doubt.
But before we get to the toxin, how do we even identify C. diphtheriae in the lab?
Well, it has some giveaways. One tool we use is Hoyle's tellurite medium.
Okay, tell me more about that.
It contains potassium tellurite, which is toxic to most bacteria.
But not C. diphtheria.
Nope. It actually reduces tellurite to tellurium.
So it basically detoxifies it.
In a way, yes. And this makes its colonies turn black.
So you can easily spot it.
Exactly. It stands out from the crowd.
That's pretty cool. But do any other bacteria do that?
Some can, but their colonies usually turn gray or brown. And to be extra sure, we use Tinsdale medium too.
Ah, yes. You mentioned that before.
On Tinsdale, C. diphtheria forms a brown halo around its black colonies.
So it's like a double confirmation.
Precisely. It's hard to miss.
And even within C. diphtheria, there are different types, right?
You're right. There are 4 biotypes.
Okay, tell me about them.
Gravis, Mitis, Intermedius, and Belfanti.
And they're all a bit different.
Yes, they have slightly different characteristics, like their colony morphology and biochemical reactions.
Fascinating. It's like they have their own little personalities.
You could say that.
But why is knowing the biotype important?
It helps us track how diphtheria spreads and maybe predict how severe a case might be.
I see. It gives us more information about the bacteria.
Exactly.
Okay, I think we've gotten to know C. diphtheria pretty well.
We've just scratched the surface.
Well, true. But now I'm really curious about this diphtheria toxin.
Me too. That's where things get really interesting.
Let's dive into that next time.
Sounds good. Welcome back. Last time, we explored the fascinating world of Corynebacterium. We talked about the different species and their unique features.
And that potent diphtheria toxin, just one molecule can cause so much damage.
Exactly. But let's shift gears a bit.
Okay.
And imagine we're clinicians facing a potential diphtheria case.
Okay, I like that.
A patient comes in with a sore throat, fever, and those grayish membranes in their throat.
Sounds like diphtheria.
It could be, but we need to confirm.
Head to the lab.
You got it. Lab diagnosis is crucial for the right treatment and to stop it from spreading.
Makes sense. So what's the first step?
We need samples from the throat, nose, and nasopharynx.
Oh.
And a sample of the membrane, if there is one.
Got it. Then what?
Get those samples to the lab quickly and carefully.
Time is of the essence.
Exactly. Once in the lab, we inoculate them onto different media.
Different media.
Yeah, blood agar standard, but we also use special ones for C. diphtheriae.
You mean Hoyle's tellurite media?
Yeah.
The one that turns the colonies black.
That's the one. It gives C. diphtheriae a real advantage.
And helps it stand out.
Exactly. But just to be sure, we use Tinsdale medium too.
The brown halo, right?
You got it. That double confirmation makes it really clear.
So we know for sure it's C. diphtheriae, but we still need to know if it produces the toxin.
That's the most important part, right?
Yeah, absolutely. So how do we do that?
There are a few methods. One is the ELEC test.
What's that?
We grow the bacteria on a special medium and add antitoxin strips.
Okay.
If the bacteria produces toxin, it reacts with the antitoxin and forms a precipitate.
So we can actually see it.
Yep. But nowadays, we often use PCR.
The go-to for so many things.
It's a powerful tool. We use it to amplify the toxin gene.
So if we find the gene, we know it's toxigenic.
Exactly. So we have ways to find the toxin directly and to look for its gene.
Like having a two-pronged approach.
Precisely. And this information helps us decide on the best treatment.
Speaking of treatment, how do we manage a diphtheria infection?
We have to do two things. Neutralize the toxin that's already there and get rid of the bacteria that's making more.
Like a double attack.
Exactly. To neutralize the toxin, we use diphtheria antitoxin.
Makes sense.
It's a solution of antibodies that specifically target the toxin,
Okay.
but it only works on toxin that's circulating in the body, not toxin that's already inside cells.
So it's like a race against time.
You got it. The sooner we give the antitoxin, the better.
So early diagnosis is crucial.
Absolutely. And we also need to eliminate the bacteria.
That's where antibiotics come in, right?
You know, penicillin and erythromycin usually do the trick.
So we hit the toxin with antitoxin and the bacteria with antibiotics.
Exactly. It's a powerful combination.
And how long does the treatment last?
It depends on the severity, but usually at least 14 days of antibiotics.
To make sure we get all the bacteria.
Right. And supportive care is important too, like fluids and respiratory support.
So it's a comprehensive approach, targeting different aspects of the infection.
Exactly. And of course, prevention is key.
Which means vaccines.
You got it. The diphtheria toxoid vaccine is super effective.
It's part of the DTaP, right?
That's the one. It protects against diphtheria, tetanus, and pertussis.
3-in-1 protection. That's great.
And it's not just for kids. Adults need booster shots too.
To keep that immunity strong.
Exactly. But what if someone hasn't been vaccinated or we're not sure about their status?
Especially during an outbreak, we need to act fast.
We do. In those cases, we use prophylactic antibiotics.
That's like a preemptive strike.
Exactly. It gives their immune system a head start.
And buys us some time to figure out their vaccination status.
Exactly. So we have strategies for individuals, but what about the whole community?
That's where public health comes in, right?
You got it. We'll dive into that in the next part.
Welcome back to the deep dive. We've been exploring Corynebacterium, say by P.
Focusing on C. diphtheriae and diphtheria.
It's been a fascinating journey, right? From its microscopic features to its impact on public health.
Absolutely.
And let's not forget those stylish black colonies with the brown halos.
Always a good reminder.
But in all seriousness, diphtheria is a serious disease, especially without vaccination.
That's right. And that brings us to the role of public health in controlling and preventing it.
So what are the key strategies here?
Well, vaccination is still our best weapon.
It's all about protecting the community, right?
Exactly. But even with vaccination, immunity can decrease over time.
So adults need those boosters.
Absolutely. It's important to stay up to date.
What about those who aren't vaccinated? Or we don't know their status?
Especially during an outbreak, we use prophylactic antibiotics.
Like a protective measure.
Exactly. It helps prevent them from getting sick while we figure out their vaccination status.
And early detection is critical too.
Right, you got it. Healthcare providers need to be on the lookout for diphtheria symptoms.
So they can report it quickly.
Exactly. That triggers a public health investigation.
To find the source and track down contacts.
You got it. It's like detective work to contain the spread.
So it's a multi-pronged approach. But how do we prevent outbreaks in the long run?
Maintaining high vaccination rates is key.
Through education and awareness.
Exactly. But what about areas with low vaccination rates or limited health care?
That's a challenge.
It is. International organizations like the WHO play a vital role here.
They support immunization and surveillance in those areas.
Exactly. But even with all our efforts, outbreaks can still happen.
So what can we learn from those?
They're valuable learning opportunities. We can study them to identify risk factors and refine our strategies.
It's about constantly improving our approach.
You got it. We need to be vigilant and adaptable to stay ahead of diphtheria.
Well, I think we've covered a lot of ground in this deep dive.
We've explored the bacterium, the disease, and how to combat it.
It's been a real eye-opener.
I agree. And hopefully our listeners feel more informed about this complex disease.
Absolutely. Now, to wrap things up, let's put your knowledge to the test with a few exam questions, just like in our source material.
Ready for a challenge?
Bring it on.
Yeah.
Here's the first one. What's a key characteristic of Corynebacterium CP? Is it A, they need oxygen to grow, B, they create spores to survive, C, they divide in a way that creates a stacked arrangement, or D, they move around using lots of flagella?
Choose wisely.
The answer is C. They divide in a way that creates a stacked arrangement. That's the snapping division we talked about, leading to those palisades.
Next question. What carries the diphtheria toxin gene? Is it A, tiny rings of DNA called plasmids, B, the bacteria's main DNA, C, viruses that infect bacteria, or D, jumping genes?
Time to think back to our discussion on how C. diphtheria gets its toxin.
It's a fascinating process.
The answer is C. Viruses that infect bacteria, those are bacteriophages, and they introduce the toxin gene through lysogenic conversion.
Question 3. Which special medium helps us identify C. diphtheria by its colony appearance? Is it A, McConkey agar, B, blood agar, C, chocolate agar, or D, Tinsdale agar?
Remember those telltale signs?
They're hard to miss.
The answer is D, Tinsdale agar. That's where we see those black colonies with the brown halos.
On to question 4. When is diphtheria antitoxin most effective? A, when the patient starts having trouble breathing. B, only after we confirm the bacteria is toxigenic in the lab. C, as soon as we suspect diphtheria. Or D, after starting antibiotics.
Timing is key here.
It can make all the difference.
The answer is C. As soon as we suspect diphtheria, we need to neutralize that toxin before it does more damage.
And the final question, what's a crucial public health measure to prevent diphtheria outbreaks? A, keeping infected people away from others. B, making sure vaccination rates are high. C, giving antibiotics to close contacts. Or D, all of the above.
This one's about the big picture.
Protecting the whole community.
The answer is D, all of the above. It's a multifaceted approach to keep diphtheria at bay.
And with that, we've reached the end of our deep dive into Corynebacterium SPP, guided by principles and practice of clinical bacteriology.
We've explored these bacteria from the inside out, uncovering their secrets and learning how to combat them.
It's been quite a journey.
It has. And remember, the microbial world is always changing, so stay curious and keep learning.
We'll see you next time on The Deep Dive.
Stay healthy, everyone.